Focal adhesions, FAs, are specialized adhesive structures which serve as cellular communication units between cells and the surrounding extracellular matrix. Analysis of FAs is critical to understand cell adhesion, which is a critical phenomenon in cancer research. Here, we have optimized the analysis of FAs in MDAMB231 breast cancer cells. The analysis steps are simplified by combining different plugins of the ImageJ program. This method is applicable to other types of image processing, such as localized or high-resolution image analysis, such as low-resolution image analysis of spatially distinct signaling structures such as FAs. Our method can be applied to many types of image analysis, such as FA quantitation, to determine how well cell adhesion relates to its biological function.
This article is titled Step-by-Step Quantitative Analysis of Focal Adhesions and was authored by Utku Horzum, Berrin Ozdil, and Devrim Pesen-Okvur.
We are article.tv. Article links can be found in the video description.